Hello and welcome to the Sound of Science. Today I want to speak with you about a topic that troubles many individuals throughout the world, and it is whether the COVID-19 vaccine affects fertility. And obviously this topic is of utmost importance to women that wish to get pregnant or women that are already pregnant, and we will try to answer all these questions. So on December 1st, 2020, Michael Yeadon, which is a former employee of the Pfizer company, along with Wolfgang Wodarg, a physician, both of them applied a letter of concern to the EMA, European Medicine Agency, stating that the COVID-19 vaccine, which targets the spike protein of the coronavirus, also can bind a protein on human cells, a protein called Syncitin 1, and they claim that this could affect fertility in women. In order to understand whether it is indeed possible that the COVID-19 vaccine can both bind the spike protein of the coronavirus as well as the Syncitin 1 protein on human cells, we'll have to understand some basic biology, some basic immunology, and actually there is no other way to understand this issue. So I will try to take you one step after the other in order to understand the basic biology behind the effects of the vaccine, and together we will understand whether their claim is actually possible. So first things first. We have to understand what is a protein. So a protein, as you can see here, is a chain comprised of specific amino acids. Here, each name that you see here is a name of an amino acid. We have around 20 amino acids in our body, and a chain of amino acids comprise a protein. These proteins are not just an elongated chain of amino acids, but these amino acids that form the protein can also form a structure in 3D that can either be this kind of sheets, or we call them beta sheets, or that they can form a helix. So this is just the way they rearrange in space. Proteins, or most proteins, are more complicated than that, and they comprise of a set of such beta sheets, as you can see here, as well as a set of helixes. So both of these forms are present within proteins and comprise the structure of the protein in 3 dimensions. This is not all, and actually proteins, or some of the proteins, can have even a more complicated structure in 3D, as you can see here in this example. Why is it important for us to understand what I just explained? So it is important in the context of vaccines. For example, there are antibodies, which are the product of vaccines, that can bind a specific set of amino acids. So they recognize specific proteins by the specific sequence of particular amino acids. So that's one type of antibodies. Another type of antibodies is the kind of antibodies that can recognize a particular structure. So theoretically, there can be two different sequences of a protein, that is, two proteins that are comprised of different amino acids, but they will have a similar structure. And so theoretically, one antibody can detect two different proteins that have the same structure. But they really have to be very identical in order for the same antibody to bind the same structure. So again, we have one antibody type that can bind a sequence, but a different antibody can bind a particular structure. So back to the same protein I showed you earlier, we can experiment with these proteins and study their structure in 3 dimensions. As you can see here, this protein has a 3-dimensional structure. This particular protein is involved in the replication of DNA. And as you can see, it's comprised of a sort of a ring, and the DNA passes through the center of this ring Actually, this protein is important to keep all the proteins that are responsible for replication of DNA tight around the DNA strand itself. This protein is called pCNA. Okay, so that was a general introduction to how proteins are built. Now let's look at the spike protein of the COVID-19 or the SARS virus. What do we know about this protein? Well, we know that this protein is comprised of 1,275 of those amino acids that I just mentioned earlier. We know that it's a fusion protein, so the meaning of a fusion protein means that it is expressed on the virus and its aim is to bind into the cell of the host, which is us, okay, the cells that are lining our respiratory system, to bind into our cells and then allow the virus to fuse with our cells and in this manner to enter our cells. This is how the spike protein looks like. This is the look from above. Now we see it from the side. Okay, we can see that it has three subunits, the orange, the purple, and the green. Now we can see the spike protein from below. But as you can see, we know a lot about the structure of this protein. Okay, this is what you see now is the result of many studies that studied the structure of this protein. Now, what does the spike protein do? So it is located around the viral body. All of these red proteins in the scheme that I made are the spike proteins, and their role is to bind to a receptor in our cells. The receptor's name is ACE2, and following successful binding, the spike protein functions as a fusion protein and helps the virus to fuse into the cells, and that way the virus can inject its inner content, and which will enable the virus to replicate itself in the cell and then propagate itself. In contrast, what do we know about the Syncytin 1 protein, which Michael Yeadon claimed that the antibodies bind to? Well, this protein is a 538 amino acids protein. It is also a fusion protein, which makes it similar to the spike protein of the virus. As you can see, we only know something like 20% of the structure of this protein. So unlike the spike protein, in which we know the structure of the entire protein, people have only studied a small portion of this protein, and it is the portion that you can see here in the video, in the animation that you see currently. This is taken from a database that all the sequences and the structures of the proteins that were studied are submitted to. So we do know something about the structure of this protein, and it is what you see right now. What is the role of the Syncytin 1 protein? So it's very interesting. Placenta is comprised of several tissue types, but one of them, as you can see here in the blue painting, is called the placental syncytiotropoblast. Okay, this is a tissue that is very important for many things. One of them is to provide nutrients to the embryo, but also it is to prevent the mother's immune system to attack the fetal tissue. And why is that? Typically, when we have a tissue that is comprised of many cells, immune cells can penetrate through the gaps that are between the cells into the next tissue. However, this tissue is comprised of one huge cell, which is the result of a fusion of many cells, and the role of the Syncytin 1 protein is to enable additional cells to fuse into this mega cell and become a part of it. Okay, so this is the role of the Syncytin 1 protein. In short, it comprises the cells in the placenta, and it is important to maintain the health of the fetus. So we are going to compare between these two proteins, and we're going to look at several aspects. One is compare the protein sequence of these two proteins. Two, we will compare the immune responses to the two proteins. And lastly, we will compare the structural identity of these two proteins. So let's start with the sequence. So as I mentioned, the SARS-CoV-2 protein is comprised of 1,275 amino acids, and the Syncytin 1 protein is comprised of 538 amino acids. Both of them are fusion proteins, so they have a similar function. And actually the fact that they have a different size doesn't matter so much for us, because, for example, these 538 amino acids can be a part of this sequence, and so therefore we have to compare the actual sequence. So let's delve into it. What you see here in front of you is the actual recipe of the COVID-19 vaccine by Pfizer. So you actually see the very actual vaccine. What you see here in the red rectangle is the actual code for the protein that will be made inside the cells after vaccination. So, and this is the code, and every 3 letters of this code are recognized by the cell as the recipe for a particular amino acid. So TTC, for example, is the code for cysteine. Okay, and in this manner, the cell knows how to read this code and to prepare a protein. So this is the RNA recipe for this protein. Okay. Now, for Syncytin 1, we also know the code that the cell can use in order to prepare the protein itself, the Syncytin 1 protein. Now, I've shown you the sequences of the proteins, and now we have to understand what antibodies do with this particular sequence. So here you can see a typical form of an antibody, and here in yellow you can see the portion of the antibody that actually binds a target. Okay, so this portion that actually binds the target, it recognizes something in the vicinity of 50 amino acids. Okay, and of those 50 amino acids, 15 of them are important for the actual physical binding between the antibody and the target. So the 50 amino acids are important for both physical binding and also some steric effects on the interaction between the antibody and the target, but 15 of those are actually important for physical interaction between the target and the antibody. Okay, so we know that. Now we want to know whether there is a sequence identity between the two proteins. I should remind you that we want to understand whether antibodies that target this protein can actually target also the Syncytin 1 protein. And our first step is to compare the sequences. Now, what you see here is the result of an algorithm that was run on the two sequences, and its aim was to directly compare the two sequences. So what you see here is a direct comparison of the two sequences. The upper sequence is the protein sequence of the spike protein, and the lower sequence is the sequence of the Syncytin 1 protein. And you can see it one line after the other, because I can't show you the entire sequence in one line, so it just shows you one after the other. And so if there is a complete identity between the amino acids of the protein, you can see it shown in asterisk, and otherwise it's in different letters. So what you can see here is that we're looking for whether there are 15 amino acids that have a complete identity between the two proteins, and we can see that there is actually none. The highest number of amino acids that are identical between the two proteins is 4 amino acids, as you can see here. And next we want to see whether there are 15 amino acids in the vicinity in order to see whether an antibody against the spike protein can actually bind also or detect also identical amino acids on the Syncytin 1 protein, and we can see that this is not the case. Degree of similarity and identity between these two proteins is very, very low. So in addition to the fact that there are no 15 amino acids that are identical, also when we try to look for 50 amino acids that will actually be recognized by the entire antibody, we also cannot find a very high similarity. And we can compare so on and so forth for the entire proteins. The degree of identity is very, very low between them. So that was a direct protein sequence comparison between the two proteins, and we found that there is hardly any identity between them. So next we need to see whether the immune system can bind similar regions on the two proteins, on the spike protein on one hand and the Syncytin 1 protein on the other hand. But we need first to understand that antibodies cannot simply bind any kind of region on a protein. Antibodies can bind particular protein sequences, and nowadays we can actually put a protein sequence in an algorithm and be able to tell which regions of this protein can be actually detected by antibodies. Okay, so that's one thing. Another thing that the immune system does is to take proteins of a pathogen, for example the spike protein, and it can digest it inside immune cells into small bits, and these small bits can then be presented on a very special receptor that is located on the immune cells. This receptor is called MHC class II, and this receptor presents portions of the original viral protein, and this presentation is done in order for other immune cells to recognize this particular protein, and it tells them that there is a virus in the vicinity of the cell. So this is the second immune receptor. However, it is also important to know that even this presentation doesn't include the entire sequence of the protein. So similarly to the case of the antibodies from plasma cells, not all the portions of the protein can be presented on these receptors, and we also are able to tell whether or which portions of the protein can be presented on this receptor. Okay, as you can see here in this example. After analysis of the regions on the spike protein and the regions on the Syncytin 1 protein that can be detected by either antibodies from B cells and plasma cells, as well as understanding which regions of the two proteins can be presented on MHC class II receptors, we can see that there is actually no match between the two proteins. So what does it mean? It means that portions of the spike protein that can be detected by immune cells are not the same portions that the immune cells can detect on the Syncytin 1 protein. Next, we want to know whether the two structures of the spike protein and the Syncytin 1 protein are actually identical in order to understand whether antibodies can bind both proteins in the same manner. Because you may remember that I mentioned that antibodies can bind both by sequence of the protein, but it can also bind to a particular structure. So we know quite a lot about the structure of the spike protein, as you can see here and as I showed you earlier. However, we don't know half as much about the Syncytin 1 protein. We only know something like 20% of the structure of the protein, and it's a region called a coiled coil domain. However, we don't know the rest of the structure of this particular protein. So any kind of claims about identity of the structure between the two proteins have no base, because we can simply not compare the two proteins. So to summarize, I showed you that there is no sequence identity between the two proteins, actually far from it. There are no common immune recognition sites between the two proteins. And lastly, I showed you that there is no evidence for structural identity between the two proteins. The question, does the COVID-19 vaccine affect fertility through affecting the Syncytin 1 protein? This is pretty much debunked because there is no actual reason for that claim. And it reminds us of the importance of staying up to date with concrete information and understanding the facts of the biology of our immune system in order to know whether specific claims are true or false. I want to thank several researchers at Bar-Ilan University that helped me prepare this video. Professor Yeadon of the Life Science Faculty at Bar-Ilan University and Professor Cyril Cohen of the Life Science Faculty at Bar-Ilan University. With that, I want to thank you. Please subscribe if you enjoyed the video. And that will be all for today. Stay safe.